जैसा कि हमने बात की कि हमारे पास 3 डिफरेंट मेथड्स हैं टू प्रिपेयर अ प्रोब। अभी हम बात करेंगे रैंडम प्राइमिंग मेथड की इन डिटेल, हाउ वी कैन प्रिपेयर अ रेडियो लेबल्ड प्रोब यूजिंग रैंडम प्राइमिंग मेथड। रैंडम प्राइमिंग मेथड में द मोस्ट इम्पॉर्टेंट थिंग इज़ द ओलिगोन्यूक्लियोटाइड प्राइमर्स। जैसा कि हमने नाम से इसको क्लैरिफाई किया दैट the primers prime at random positions. So the primer should be a mixture of all possible sequences which can bind to the template DNA. We can say this is a heterogeneous oligonucleotides which forms hybrid with the template strands. When we talk about radioactivity, then again we need a nucleotide which has its alpha phosphate radiolabel P32. It can be DATP, it can be DCTP, or it can be more than one radiolabel nucleotide, and 2 nucleotides which are normal nucleotides to generate the probes. If we compare the random priming method with Nick translation method, the major difference is in enzyme activity and the product size. रैंडम प्राइमिंग मेथड में हमें किसी किस्म की एक्सोन्यूक्लियस एक्टिविटी की ज़रूरत नहीं होती, वेयर एज इन केस ऑफ निक ट्रांसलेशन 2 टाइप्स ऑफ एक्सोन्यूक्लियस एक्टिविटी इज रिक्वायर्ड, दैट इज डीएनएज 1 एंड 5 to 3 एक्सोन्यूक्लियस एक्टिविटी। अब ये एक्सोन्यूक्लियस एक्टिविटी अचीव की जाती है डीएनए पॉलीमरेज 1 एंजाइम फ्रॉम ई कोलाई यूज जब किया जाता है। ऑन द अदर हैंड random priming के लिए we only use the Klinov fragment which have the polymerization activity, not any kind of exonuclease activity. So यहाँ पे हम ये claim कर सकते हैं कि random priming method generates the probes only by extension of the primers. Random priming का जो दूसरा benefit है हमें, the size of the probe is homogeneous and we can control the size of the probe which is radioactively labeled. On the other hand, when we talk about Nick translation method में we have no control on the length or the size of the radio labeled probe. When we talk about the components required for random priming reaction, we need a template, DNA template, we need a polymerase, we need the radio labeled nucleotide and we need the primers. जब हम polymerase के हवाले से बात करते हैं, we are going to use Klinov fragment from E. coli DNA polymerase 1 because of its ability to work under different sets of conditions. On the other hand, the other polymerases, they may have some limitations with their specific activity of polymerization. The radioactivity or the radioactively labeled probe by random priming method is only going to be available by extension of primer instead of replacement of any other existing strand. अगर आप इस पिक्चर को देखें, this shows you the template और हमारे पास जो प्राइमर्स हैं दे आर रैंडम प्राइमर्स, दे बाइंड एट डिफरेंट पोजीशंस. अब Klinov fragment is going to extend this end of the primer and is going to stop at this point. Similarly, the other primer is extended and in this way all the primers are going to be extended to prepare this new strand which contains most of the radio labeled nucleotides. So this is the extension of primer, not the replacement of any DNA fragment. Replacement का मतलब ये होगा कि एंजाइम इस पर्टिकुलर पॉइंट से ऑनवर्ड जितना भी आपके पास न्यूक्लिक एसिड स्ट्रैंड है, वेदर दिस इज द प्राइमर और दिस इज द एक्सटेंडेड स्ट्रैंड, इसको एक्सोन्यूक्लियस एक्टिविटी के जरिए रिमूव कर दे और इसकी जगह पे नया स्ट्रैंड सिंथेसाइज करे. So Klinov fragment is only going to extend the primer and is going to give you the probe. Radioactivity is achieved by radiolabeled nucleotide and this is because of phosphate, 3000 curie per micromole should be the specific activity of that particular nucleotide. Most of the time we use DCTP as the radiolabeled nucleotide, but sometimes we have to use DATP as radiolabeled nucleotide. The other three nucleotides are the normal one which are available from commercial sources. During the synthesis of the new strand, अगर 40 से 80% तक radiolabeled nucleotide incorporate हो जाता है newly synthesized strand में, that is sufficient enough to give very good activity. And if we talk about the rate of incorporation, 1 out of 25 to 35 nucleotide is always the radiolabeled nucleotides incorporated in the newly synthesized strand. Primers are very important. The length of the primer plays critical role. In this case, we use a primer which is 6 nucleotide long. अगर shorter nucleotides length के primers use करें, they are very poor primers. And if we increase the length from 7 onwards, then there are problems of self-annealing. So the hexamer indicates that the length of the primer should be 6 nucleotides long. Then the concentration of primer matters. Very high concentration or very low concentration, both are not good. In case if the primer concentration is very high, it hinders the process and it is responsible for exhaustion of the radiolabeled nucleotide. Very low concentration of primer is going to give you heterogeneous lengths of the radiolabeled products. As far as template DNA is concerned, the shorter is the template DNA, very few nucleotides will be incorporated and we are going to have problem in the specific activity. If this is a closed circular DNA, double-stranded DNA, then it is not a good template. It should be converted into the straight chain or linear molecule with the help of endonuclease enzyme. Wherever practicable, always use purified DNA fragment to prepare the probe. It is beneficial because जब आप probe prepare करते हैं और अगर आपके पास required DNA fragment के अलावा fragments होंगे, then we are going to suffer from high volume or high level of background when we are going to use that particular probe for labeling or for southern blotting. So purified DNA sample is advisable to use for probe preparation.